I have a sign-up sheet on my bulletin board if you have questions. Guarantee yourself a space sometime. Would be good. And we can kick everybody else out and say, oh, yes, I have it. So sign up. What else do we need to talk about? Oh, you have an invocator thing due tomorrow at 5. To get that off your plate. Get that off your plate. Okay. This afternoon I will post, I already have a PDF copy of my solutions to the practice problems, and I will get them on Scholarly this afternoon. So if you please solve the problems and take a look. But don't look too quickly, okay? Chew on it for a bit. So Rachel, I had a little bit of issues yesterday uploading, so the part 3 is going to take a while. Okay, and I think what I'm going to do is, Shia is gone. He's getting the George Washington Carver Award in St. Louis, so yeah, I think that was cool. So he's gone today and tomorrow, so I'll probably continue to record and get them uploaded as fast as I can. So I might leave my computer here tonight to do the rest of the uploading, because all I got to get the biochem lab report graded. I don't need a computer for that. Okay, so any questions about what we did yesterday? Okay, all right, so today we're going to talk about the sodium-potassium pump, and then we'll talk about a calcium pump. Okay, so that's probably the common term, right? Is that how you guys, you've called it or named it in the past? Okay. Also, it could be called the sodium, potassium, ATPase. Because it is an ATPase. Okay, so I wanted to take a look a little bit. I'm going to go back to KMs. I'm going to think about KMs. Oh yeah, but first of all, I want to tell you why we use the sodium-potassium pump. The predominant function throughout the body is to control cell volume. Okay, so that cells don't swell due to all of the stuff that they accumulate in the cytoplasm. The movement of more ions out of the cell than into the cell can shift the balance of water movement a little bit. Okay, so we've also seen, and I think more, some of you have had physiology. Is that we talk about the sodium-potassium pump as restoring the membrane potential or the voltage across the membrane after an action potential or even a series of action potentials. Okay? But that has use really for electrical potential, for action potential, both in the muscle and in the nerves. But we use the sodium gradient for active transport. And probably all cells use the sodium gradient for active transport. It can be used to regulate pH, so because you can exchange a sodium ion for a proton, you can allow a sodium ion in and pump a proton out if you need to get rid of some hydrogen ions and raise the pH a little bit. Okay, and regulates urine volume. Okay, so if you need to preserve the amount of fluid that you are circulating, you could activate the sodium-potassium pump to move more sodium back into the bloodstream, and that'll draw the water back into the bloodstream. So aldosterone controls the sodium-potassium pump. So when you are dehydrated and you've lost some body fluid and need to get the blood pressure back up, aldosterone can alter the activity of the sodium-potassium pump. Yeah. Oh, and I'll tell you at what step, okay? When we get there, I'll tell you what step.
Does that cause more sodium to go into the blood or more potassium to go into the blood?
Well, let's think about that. That's a good question. I'm just so, look. Okay, so there's the apical, this is a kidney cell, by the way. You didn't recognize what this block is? It's a kidney cell, okay? And the sodium-potassium pump is down here in the basolateral side of the cell, okay? And when the sodium-potassium pump is operating, where is sodium going to go? Always goes the same direction. Up. And 2 potassiums are pumped. Okay, so what could happen to the amount of potassium if you are activating this pump?
Increase.
Yeah, you could lose some potassium. Yeah. Okay, how about this? And then with sodium being pumped out of the kidney cells, so and back into the bloodstream, the water will be drawn in, or will at least not osmose into the urine. Okay, so aldosterone lowers blood volume predominantly, well, one way is by activating the sodium-potassium pump. Okay, so this is, aldosterone, well, I was going to say, there are some diuretics that cause the loss of potassium, but that's a whole other story. Okay, are you ready? Oh, I think I got everything, yep, I think I got everything listed. So it is the most common, sodium-potassium pump is the most common plasma membrane protein in your body. All cells use it for one of these functions, and really active transport is a pretty standard function for the sodium-potassium pumper to maintain that sodium gradient. Okay, it is a P class of ATPases, or as I ended up calling it an E1, E2 ATPase. Okay, so we're going to start looking at the conformation in E1, and of course my pictures are highly schematic and anything but realistic. So, but at least it gives you the idea of what in the heck is going on. Okay, so probably my proteins are going to grow as we move across the board. So we're going to start out with the E1 conformation. Okay, the E1 conformation has 3 binding sites for sodium ions inside the cell. Okay, so here's out again, and here's in. Okay, so I'm going to I'll try to draw use red for sodium. The Km, because this now you can measure, in this case you can measure the Km because we're not using cooperative binding. So the binding of one sodium is not going to influence the binding of the other sodium ions. It's really small, 0.6 millimole. Okay? But inside the cell, the sodium concentration is about, we'll just use what we've been using, 12 millimolar, okay? So does that concentrate, the difference in concentration and Km, is that gonna facilitate the binding, or is it gonna make it really hard for sodium to bind? Is it gonna be easy for sodium to bind or hard? It's gonna be easy, okay? So we're operating the real concentration of sodium inside the cell, even though this is where the sodium concentration is low, it's still way higher than the Km. This is an extremely low Km, okay? So that's gonna facilitate the very low Km for sodium is going to facilitate the binding. Of sodium. Okay, when sodium binds, it activates an ATPase activity. And I am going to summarize this again. So don't get it all down in words. I'm going to summarize it again in a different way, so you can see the involvement of sodium and ATPase.
Is sodium still bound?
Yeah, sodium's still getting bound, but not for long, because there's going to be a conformational shift. In the sodium, potassium pump. So the conformational shift pulls the sodium to the outside of the cell, and we lose the affinity for sodium. Okay, and if you remember, we've been using the sodium concentration outside the cell in 145 millimolar. Okay, so the E2 conformation has no binding site for sodium, and so sodium gets deposited on the outside, even though there's a pretty high concentration of sodium outside the cell. Okay? E2 conformation has a fairly high, actually a pretty darn high concentration, or Km for the binding of potassium, which we'll do in red. So potassium would be we'll take a look at Kms. So outside the potassium binding site, the Km is 0.2 millimolar. That's a high affinity binding site. And the concentration of potassium typically outside the cell is 4. I think we've been using 5 to 4 millimoles. Okay, so again, this extremely high affinity binding site for potassium favors the loading of potassium even at really low concentrations of potassium. Okay, so potassium binds, and when potassium binds, it's going to stimulate the release of the phosphate group. Okay, and then the conformational shift back to E1. Is going, that shift pulls the potassium in, but then we lose the binding site for potassium, and we shift back into E1 conformation, and you can start the cycle over and over again. And the sodium potassium pump, you do about 100 cycles per second. Okay, that's, to me, rather amazing how fast that can happen. So you can move 300 sodium ions out of the cell per second, 2 potassiums into the cell, 200 potassiums into the cell per second. Okay. Let's see. This step here, the ATPase activity that helps convert E1 into E2.
So the addition of the phosphate group and then the subtraction of the phosphate group is what causes the conformational change?
Right. Right. And you can only phosphorylate the sodium potassium pump when sodium is bound. And you can only remove the phosphate group when potassium is bound. Okay? And the phosphorylating event here is accelerated by aldosterone. Okay, aldosterone enhances the rate of ATP hydrolysis. Okay, so I'm going to summarize this a little bit differently.
So like the first phase, the inorganic phosphate, is it from the hydrolysis of ATP?
Oh,
They
are you
want
asking
that-
about aldosterone?
I mean the phosphate that gets bound to the protein then, is it from the inorganic phosphate?
Right, so you go from adenosine triphosphate, you cut one phosphate group off the ATP, put it on the protein, and then what you have left over is adenosine diphosphate. And we've actually, and I'll come back to this because I'm going to rename this enzyme, this step. You could call this step an ATPase, but that's not the best, I think, description of that enzymatic activity. So we'll get to a different term. So E1 fluctuates, this carrier fluctuates between two conformations. One, this really requires 3 sodium ions. ATP is hydrolyzed, and inorganic phosphate's attached. And the amino acid that is phosphorylated is not your typical amino acid that gets phosphorylated. So I think that in itself is going to be. Okay, this is an aspartic acid. So the phosphorylated amino acid in the sodium potassium pump is an aspartic acid. E is a single letter abbreviation for that. And it looks like when in the phosphorylated form, it looks like that. Which is, can you imagine what an electrophilic core that is? Get 4 oxygen, 2 of them are charged. And I can't even imagine what gets attracted to that. Okay, the removal of the phosphate group requires 3 potassiums. And the phosphate's taken off inorganic phosphate. So the whole of the reaction is we're seeing ATP is hydrolyzed to ATP plus PI in really two different steps, right? So this is the sum of these two reactions. Okay? This reaction is actually a really nice protein kinase reaction. Okay? So in a protein kinase reaction, a protein gets phosphorylated, and ATP is always the source of the phosphate group. So this is a classic protein kinase reaction. We will see, so from here on out, from I think probably Unit 2 to the end of the semester, we are going to encounter so many protein kinases that if you can't think of a right answer for on a test, I'm going to say, what's the function of this protein? If you can't think of the right thing, say protein kinase, and you're probably right. Okay? I always, and I'll say that again, but that to me, my default answer in organic chemistry was steric hindrance. Have you guys ever tried that? You just don't know what the mechanism is. Well, steric hindrance is always a good shot. Okay, so proteins, that enzymes that take phosphate groups off of proteins are phosphatases. Okay, so this is called, so the sodium potassium pump has protein kinase activity and it has phosphatase activity. So the overall sum is that it is an ATPase. So just to summarize, we saw that aldosterone is going to facilitate this step. But digitalis is going to block, it's going to block that step, okay? So in the sodium potassium pump, in the presence of digitalis, can become phosphorylated to form the E2 conformation, but the phosphatase activity is blocked. So the protein becomes stuck in the E2 conformation. There's another protein that, another drug that's always been used in research, has the same function. I always spell it wrong too. Just a second, I can't remember if the O or the U comes first. Okay. O comes first. The drug Wabi. I don't think that's ever been given to patients, but it's used a whole lot in research. Whopping works like digitalis. It knocks out the phosphatase activity. So you can study what happens if you knock out the sodium potassium pump by giving cells growing in culture some whopping. Okay, I don't think there's any other nifty details. I think that's it. All right, so that takes care of that. Like that's like a really sketchy view of the sodium potassium pump. I find it again fascinating that KMs concentrations are really driving the movement or driving the reaction in a particular direction. Okay, I don't think in physiological conditions do you ever reverse this pump to make ATPases. I can't imagine what the mechanism allows it. Okay, so you're ready to move on to one more ATPase. One more, and that's it for this unit. Yeah, okay. And then tomorrow will be all review. You guys can control tomorrow by bringing questions. I'll have questions for you, but don't rely on me to keep you entertained for an hour with questions. So please bring your questions. Okay?
quick question.
Uh-huh,
On your study guide, you have cardiotonic steroids.
that's digitalis.
Okay.
Because I didn't mention digitalis, but that digitalis would belong to that class of drugs.
Would does Wabin belong there too?
I don't know. I don't know. Actually, I've never looked at the structure of either one of those molecules. Okay, so who has not had physiology? I'm gonna try to be super friendly. Oh, a bunch of you have not had physiology yet, right? Yeah, okay. All right.
Too late.
Gee, you should have. No.
Totally true though, it's too late.
Okay, so I'm gonna draw the, this is a sarcoplasmic reticulum.
All right. Okay, so I'm going to add R. Sarcoplasm. Reticulum. It's essentially a sack of calcium. It probably does other things like manufactures lipids and things for the skeletal muscle, but for now it's a sack of calcium. And a lot of calcium also. Okay, my notes are scattered all over the place on this because I've never really put together the two story. Ah, there it is. Okay. Oh, no. Okay. So muscle contraction it requires the movement of calcium, or the presence of calcium so that actin and myosin can contact each other and do that sliding filament thing. Muscle relaxation requires, you've got to get rid of the calcium if you're going to release the actin from the myosin. Okay, so muscle relaxation in the cytoplasm, okay, we have to have a low calcium concentration. Okay, that's the only way your muscles can relax. You can release the actin from the myosin. In order to get the actin to bind to the myosin, okay, we've got to raise the calcium concentration, and it raises about 100-fold, as you'll see, because I'll write down some of the changes in the concentrations. Okay, in order to raise the concentration of calcium in the cytoplasm, here's a calcium channel that opens up. This is a voltage gated. Calcium channel. Okay. Meaning that the nerve impulse that stimulates the muscle changes the voltage in the muscle, and this calcium channel opens up. Okay, and the muscle can contract. Okay, to relax the muscle, you have to get rid of that calcium. And so we'll use a calcium pump that really is like the sodium potassium pump. It's a P class of pumps. It has an E1 conformation and an E2 conformation. So it's that protein, both the protein kinase and the phosphatase. And that's how we get the calcium back into the cell. Okay, the name of the protein is circa. And I don't know if anybody's in biochemistry has ever looked at circa. You guys ever think I can talk about that? Okay. It's a typical biochemistry, protein and biochemistry to study because its mechanism is so well known. So it stands for sarcoplasmic or endoplasmic reticulum calcium ATPase. That's circa. We'll look at a little bit of how it works. Okay, so what does the cardiac muscles do use circa, but because they don't have, the cardiac muscles don't have a whole lot of sarcoplasmic reticulum, the cardiac muscles are also relying on that calcium sodium symporter. Okay, so the cardiac muscle cells are a little more special. They have to have 2 calcium pumps, but skeletal muscles, there's enough sarcoplasmic reticulum, enough circuit to remove a sufficient amount of calcium from the cytoplasm.
So during relaxation, the calcium concentration in the cytoplasm is low.
Right, yeah. So you want to, it's going to always, the concentration of calcium in the cytoplasm is always going to be much, much lower than in the sarcoplasmic reticulum. You're never ever going to empty this guy. Okay, are you ready to look at the pump? Okay. I'm going to draw a couple of concentrations. So at rest, let's do this. Let's do this. The calcium concentration in the cytoplasm at rest is 10 to the minus 7 molar. Okay. So what is that? 0.1 micromolar.